Hi everyone, welcome to the poster scConsensus, combining supervised and unsupervised clustering for cell type identification in single-cell RNA sequencing data. My name is Florian, I am a postdoc at the Genome Institute of Singapore, and this is joint work with my colleague Bobby Ranjan. So when we cluster single-cell RNA sequencing data, we usually have the choice between unsupervised methods and supervised methods. Unsupervised methods are really great to find novel cell types and cell states, and cell types are usually annotated using differentially expressed genes between clusters, literature markers, and canonical marker genes. Using supervised methods, we have a very strong robustness against batch effects, but we do have to have a reference and might be a little bit limited by that particular reference. So we thought maybe there's a way to combine the good properties of both of those methods into a unified consensus approach. This is exactly what scConsensus is about. So as you can see in the middle, in the scConsensus workflow, we consider two input clusterings, a supervised clustering and an unsupervised clustering. In theory, you could also have two unsupervised or two supervised inputs, or actually even more. But we have shown in the paper, which the link you can find below at the poster, that actually the combination of exactly two methods, and those two methods being a supervised and an unsupervised method, is giving the best results. So having those two input clusterings, we form a contingency table, and then using this contingency table, we generate something we call consensus labels. So these consensus labels are nothing else but identification for the cluster identity a particular cell is assigned to by both input methods. And using these consensus labels, we go ahead and call DE genes between all consensus clusterings, and we use these DE genes as feature genes to recluster all our cells in this consensus space. And this is actually where we really can see the improvement of the merging of the two clustering results. So if you say we can see improvement, it means we have to benchmark the clustering performance, and we cluster by using two different kinds of data sets to obtain a ground truth. We use site-seq data as well as fact-solid PBMCs. For the site-seq data, we have 5 different data sets where we obtain a ground truth using antibody-derived tags, ADTs, and this is shown in the figures here A to E, and then we compute the normalized mutual information between the clusters we obtain in single-cell RNA-seq space to those ADT-based labels. And we have used here two unsupervised and two supervised methods, singular, RCA, scanpy, and SURAT. And then we have looked at those 5 different data sets, and what we found on average that we get the highest NMI, the highest normalized mutual information between the single-cell RNA-seq clustering and the ADTs by combining exactly one supervised and one unsupervised method. So this really leads us back to our hypothesis in the beginning that it's really useful to combine unsupervised and supervised methods because they do have quite distinct different aspects. And another anecdote, if you want, that illustrates this is shown on the right side using the facts data set. And here the important thing are this regulatory T cell population shown here at the bottom, and the labels for the UMAP. And if you look at the F1 score for cell type assignments, you can see that those regulatory T cells, they were picked up by RCA, a supervised clustering method, but they were not picked up using unsupervised clustering with SURAT. And the interesting thing is that when we use scConsensus to unify and to combine RCA and SURAT clusters, we get a very strong and pronounced population of those regulatory T cells. So this really shows again how powerful such a consensus approach can be. And if you have any more questions about scConsensus, please leave me a question. I'm happy to answer it. You can also have a look at the preprint, at the published paper in BMC Bioinformatics. It's not a preprint anymore. You can also go to the GitHub and just download the R code and try it yourself. Thank you very much for visiting the poster, for watching the video, and enjoy the conference. Thank you very much. Goodbye.